Okay. Good morning, everyone, and thank you for joining today and the Horizons in Genomics seminar. It is my great pleasure to introduce today Professor Richard Gibbs. Dr. Gibbs, he did his bachelor's and PhD at the University of Melbourne in Australia. And then he went to Baylor College of Medicine to do his postdoctoral training at the time at the nascent Human Genetics department at Baylor. And at the time where really Human Genomics was starting to discuss all of the plans for the Human Genome Project. So he became the founder and director since of the Human Genome Sequencing Center at Baylor College of Medicine. And he's been one of the main players of the Human Genome Project and human genomics over the last several decades. So I'm really happy and honored to have Richard here today. And Richard, we are very excited to hear about your perspectives on the last 3 decades of the Human Genome Project and now to precision medicine. Thank you.
Thank you so much, Claudia. Nice to see everyone, and I hope you can hear me clearly. Let me set up some slides here. I'll just get my sharing going, and hopefully now you can see the full screen.
Yes.
That's working?
Yes, now we can see. Yeah.
Excellent, thanks. So please interrupt me at any time if you wish. It's a really special time to talk to you because last month we celebrated the 20th year anniversary of the completion of the Human Genome Project. That was a 13-year project. And so we've been reflecting a lot in the last months about what it all meant and how we got here and what this means as we move ahead into the future. So that's how I want to frame my comments for you. See if I can get the slides to advance here. There we go. I need to disclose that I don't have any disclosures that are very significant. No conflicts of interest, really. So in thinking about the Genome Project, I want to tell you a little about its history and how we participated in that here at the Human Genome Sequencing Center at Baylor College of Medicine in Houston, and where that's led us to in terms of technical development and the state of genetic discovery, and what's really important to do right now, given where we're at in genetic discovery. Now, as you know, I think when the project was beginning, I suspect many of you were in grade school. I don't know, you can tell me where you were in 1985, probably not thinking too much about genetics. So it's a long time ago. The project came with enormous promises. The idea that the genomics would somehow be the 21st century genetics, and that instead of all of the sort of soft markers for genetics, we'd have a digital future that the DNA sequence would mark. And that we'd use this to understand disease mechanism, and we'd build tools and ultimately be able to understand disease better and build new treatments based on this knowledge. So here's the timeline of the program. As I said, many of you are probably not doing too much back in 1985. That was the pre-Human Genome Project period when there were discussions leading towards it. The actual project began in 1990 and took 13 years, reaching its completion 20 years ago in April of 2003, and then was followed by a period of technical development, leading us to now to our present period of personalized genomics and precision medicine. Those very early days, I won't talk too much about them, but if any of you are particularly interested, there's some excellent writing, particularly by Robert Cook-Deegan on the origins of the program. But some sort of historical moments were when the Department of Energy in the US, who were very interested in radiation effects after the atomic bombing in the Second World War, they interested in big science. They came together to say that if they had the genome, it would be useful. Over the other side of the slide here, Renato Delbecco said before, in about 1984, said, you know, if we had a genome sequence for a cancer cell, we really understand a lot more about cancer. And then Sinsheimer and Watson and others began to pull the program together with different meetings and getting funding from the US with the help of favorable legislators, and managed to get the project going. Now, remember that at that time, the promises kind of fell into two categories. One, listed on the right here, are kind of the promises that I already mentioned, the idea that the project would be a very good way to make genetics more digital, that there would be technical innovations, ultimately cost savings, that we'd have the comprehensive view of the genome that we never had before, and that there'd be computer resources that would be built around the program. And this was altogether make genetics and much of science a different way of doing things. But if you read some of the old literature, you'll see there are also some very hyped up promises: personalized medicine, new drugs, intelligent therapy, cures for cancer, the secret of life, design of babies, population health, all kinds of things that were maybe a little more hyped. So with that all going on in the background, in 1990, the National Institutes of Health formed a new Genome Center, and they had James Watson be the head of that. He was displaced shortly after by Francis Collins, who I'm sure you all recognize as the individual who really coordinated and led much of the program. I already told you it took 15 years. It cost about $3 billion. About 500 million of that was ultimately the sequencing. And at outset, it was decided that model organisms should be sequenced as well as humans, E. coli, Drosophila, worms, Arabidopsis, and yeast. Some of the money should be spent on ELSI, social and legal issues, and that the product should be of high quality, because if it was going to be ultimately usable, it needed to be of high quality. Low quality data would slow up everything downstream. And that became an important thing as the project moved ahead. Now, at the outset there, we realized that we needed technical development. And I think this is one of the big messages of the program. Very often, I like to say science is driven by technology, and I think most people agree with that, but many people develop technology and then ask, what can I do with that technology? And what was remarkable about the Genome Project, a little like the Moon Project, was the goal was settled before the technology was there. We said, this is where we want to be. We have to go and develop the methods for that. And this is a good principle to follow in science, I think. So, we have a question or just a beep? No, just a beep, I think. So many of you won't be able to realize how bad things were back in the 1980s from a technology point of view. And up in the top right of the screen, you can see pretty much what things looked like. There was no polymerase reaction, and I hope you're sitting down. There was no computers, no cell phones, no internet. It was almost unimaginable. In fact, our DNA sequencing was done with radioactivity, and we were celebrating then that we'd gone from Maxim-Gilbert sequencing, which is where very toxic chemicals had base-specific cleavages, through to the Sanger method, where enzyme extension was made possible by the utilization of dideoxynucleotide terminators, where the DNA sequence was terminated to make chains. But even with that method, you could get a whole thesis by dissecting one gene and doing a bunch of sequencing. And I know you've heard this story before, but that's what it looks like. So things were pretty bad, so we looked for new technologies to advance the cause. There were some pretty wild things that were suggested. Here's my favorite. It was the idea that perhaps you could take a single molecule of DNA and dangle it in a tube. This would be the tube, and you could flow past that single molecule a solution with nucleases in it that would nibble away at the DNA chain and release bases one at a time. And then with mass spectrometry, you could then go ahead and see which base was released. Great idea. It was never made to be practical, unfortunately. Others wanted to use microscopy. And this is a kind of low resolution picture. these are the bases here, but higher resolution pictures were suggested to be able to get actual DNA sequence, mainly using scanning, tunneling microscopy. Even this failed ultimately, and at one point, somebody published a paper saying they had DNA sequence, but the secret was they had never put DNA on the slide that was analyzed. So it just didn't work. Others wanted to use oligonucleotide hybridization to interpret the entire human genome. And the idea here was if you had a library of short oligonucleotides of known sequence and hybridized them to different parts of the genome, you could, from the successful hybridizations, deduce the... And this would work out if you had an enormous library of oligonucleotides or perhaps some other tricks to make the hybridization possible. Unfortunately, the math just didn't work out. Now, here's my favorite, one of the, another favorite, I should say, George Church. I'm sure many of you know his name, who suggested that in order to do this enormous amount of sequencing, we could clone, first of all, genomic DNA into many different vector libraries, many different vectors, and then combine those into a pool, and then do the sequencing on the pool, and then transfer, after the sequencing reaction was run on a gel, onto a nylon membrane, and then hybridize that membrane with probes that would light up the individual clones from the individual vectors. Excuse me. So this is the kind of picture you would get from a sequencing gel, but you'd be able to hybridize 40 or so times this membrane and reveal the DNA sequence. It wasn't practical. It went quite a way, and I couldn't find the pictures, but the machines looked like giant cement mixers that you could do these, put these membranes in and automatically wash them. So around the mid-80s, Lloyd Smith in Leroy Hood's lab published this quite spectacular paper, which really led to the technology we ultimately used. And that is the idea that if you put fairly recognizable fluorescent dyes onto different DNA primers, then you could label sequence reactions and then run them on a gel, and then using a laser detector, read the fluorescence, distinguish the four bases, and have a DNA sequence. And this is now what you call a somewhat familiar ABI sequencing machine that did slab gel electrophoresis. So we jumped on that here in Houston and started doing fluorescence sequencing and built our own little mini factory right as the project began. But we were particularly inspired by the fact that once we had a little bit of genomic sequence, we could use PCR to interrogate particular genes very effectively. And this is one of the, in fact, the first time that a fluorescent DNA automated sequence plus PCR was used to detect a heterozygous position from a woman who is carrying a genetic disease. So we rationalized that if we did this for all genes, and we could really change genetics. So we wanted to use fluorescence sequencing to work the Human Genome Project. Now, of course, others were doing this. The Worm Group, Bob Waterston in St. Louis and John Solston in the United Kingdom were working on the worm genome. Lee Hood was sequencing the T cell receptor, and Craig Ventnor was doing ESTs and other neurogenes while we were doing our work in the human. But this is where automated sequencing really got its first traction in the Genome Project. Now, by 1995, so a couple of years after the project had officially begun, the automated sequencing finally got its sort of full recognition as the method that would probably be used to go ahead to do the rest of the program. And we began to hear very much less about other radically different sequencing methods that could impact the program. And if you did an assessment in 1995 of how much DNA had been sequenced and put into public databases, almost all of it was from automated fluorescent DNA sequencing. I think only the retinoblastoma gene had some radioactive sequencing in it. So there are many editorials at that time that said, let's get on with it. We've been doing the Genome Project for a few years now. We really need to get going and begin the sequencing. So that was the technology to do the actual DNA sequencing. But the question is, what would be the strategy to sequence a human genome? Now, there are a few things that happened around that moment that formed the strategy discussions. First of all, the labs sort of focused down on a set of individual groups that were really intent upon getting the genome sequenced. All in all, at that time, there were about 20 labs. There were 6 National Institutes of Health labs, including ours in Houston, the Department of Energy, the Sanger Center in the UK, and labs in Japan, Germany, France, and China. So we all got together and we allocated chromosomes to different people. We adapted to changes in the chemistry because at that time, the idea of putting the fluorescence on the primer became obsolete by the development of fluorescent terminators. And you can see the structure of one version of the terminators with the different dyes residing on the base and that molecule being a terminating molecule. But so with that in mind, we debated the strategy of sequencing a genome. Now, you all know now about whole genome shotgun, but in those days, that methods for that were very crude. We were using a clone by clone strategy, and I'll call this plan A, because you'll see in a moment that we actually switched this. But the idea of a clone by clone strategy is that you take the large genome, 3 billion bases, and you shear it down to manageable size pieces, about 100,000, and then you do your best to order these clones by mapping and some hybridization experiments, so you get a rough idea of how those clones relate to each other. And then you take them one at a time and you shear them up some more and do random sequencing on those. And these red arrows here depict a regular sequence read that's meant to cover this, this individual genomic clone. Now, after you've done enough of that, and at that time we did 7 to 10-fold coverage, you could use a computer to assemble the product and you'd get reconstruction of the original 100,000 base clone with some regions that were not perfect and with some errors along the way. Now, this we call the draft sequence, and it would be 98, 99% accurate. But to drive that then to the high quality product that we all decided that we wanted took a lot of extra time and cost and additional reactions. So these were called finishing reactions. And the idea was you'd make individual technicians would take one of these assemblies, make oligonucleotides, and then do additional sequence reactions and additional computer work, and then produce what we call this essentially finished clone. This would be the essentially finished clone. Joining them together would give this essentially finished genome. So that's the plan we set out with in about 1997, having refined all the components and developing the technology. Now, we had many, many meetings through that period to discuss sequencing, and some of those meetings were in nice places. One of them was Bermuda, and this is the origin of what you probably recognize today as the Bermuda principles. The idea that you should have free data release of genomic data wherever possible. That idea was actually brought up in one of the first Bermuda meetings and embraced by the sequencing community, and that's been what you see propagated to today. Now, at those meetings, we also ratified the idea that we wanted a high quality genome. So when it was suggested at one of those meetings that we do a whole genome shotgun, we were all, after debate, we decided that the whole genome clone by clone method would be superior, and that we shouldn't at that time jump to doing a whole genome shotgun. Now, the idea of a whole genome shotgun, of course, is that in contrast to the clone by clone approach, you simply take the whole genome of all 3 billion or 6 billion letters and shear that and get as much random sequence as you can, and then rely on a computer to put it all together across the whole genome. Now, this is Jim Weber, and he came to the meeting and he had developed this idea with Gene Myers based upon early work done by Craig Ventnor in bacteria that showed that whole genome shotgun sequencing would work in bacteria. And there's an interesting paper you can read in Genome Research with the pluses, with an accompanying paper that discusses the pluses and minuses of that kind of approach at that time. In any case, we decided that that wasn't the way to proceed and that we would stick with our clone by clone method because it adhered to the idea of giving high quality genomes. Now, what we thought didn't matter so much, unfortunately. And what had happened in that period is that Applied Biosystems, who manufactured those machines, had developed the new generation of instruments that used capillaries. Instead of loading the sequences on a slab gel, probably many of you don't know what slab gels involve, but they're quite cumbersome. The new machine would put the sequence down through a thin tube. With Mike Applied Biosystems, they started a new company called Celera, and Craig Ventnor was to be the head of Celera, and its mission was to generate a whole genome shotgun of the human genome. Now, this was a little alarming to us in the public sector because there was no commitment of that model for a free data release. So Celera might indeed be able to do some kind of whole genome sequence of the human, but the data would not necessarily be freely available, and we felt this would be a big impediment for international researchers. Now, it was some quite feisty discussions, some quite heated discussions, but in response, the Wellcome Trust, who support the British research effort, they doubled immediately their commitment to the genome. This is in early 1998. And the National Institutes of Health also changed its strategy and focused its activities in 3 centers. That's here in Houston, also in Boston, and also in St. Louis. Plus the Department of Energy joined in. Not only did the effort become more focused, but we altered our short-term strategy. So the dilemma we had is that the non-public version of the genome proposed by Celera could be released and information could be got from it, but it wouldn't be as good as it was needed for its ultimate utility, but a lot of people wouldn't know the difference. So what we did is we altered our strategy to be more in line with what was being suggested with the development of the new instruments and the activity from Celera. So this is that same diagram, we'll call it Plan B now. Take the genome, divide it up, do this random sequencing phase, but instead of immediately finishing it, we would defer the finishing and we would join together these partially finished clones, make a draft genome, and much later, as we could over the period of time, we would finish and put in our finishing effort. And this product would then be comparable with the Celera product, but would be deliverable on the same time frame. So moving through 1999, we had altered strategy, we ramped capacity, and we did manage to generate a draft from the human sequence. At the same time, Celera did its whole genome shotgun. And in fact, as the history books will say, we all finished those draft genomes at about the same time in 2000. And here, the public group at meeting at Baylor discussing the strategy and how to do that right after the Celera announcement. You can see Francis here and Eric Lander, that's myself, and George Weinstock, John Solston. And here is Francis and Craig at the White House celebrating the joint completion of the effort. So we published at that time, producing some early insights into the genome structure, as did Celera, but we also produced a new phenomenon, which we would call the consortia authorship. You can see the many authors that were part of that paper. So that was the trip to the White House in 2000, celebrating the joint completion of draft sequences of the genome. But because of our commitment to quality, we said they were not finished, and we went straight home from there and kept on working to sequence individual clones and bring them to completion. That required many gaps that we required. There were thousands of gaps in that draft. We fixed most of those. There was new sequences came along, new chemistry came along. This sequencer that was developed during that period. And we kept at it, and by 2003 completed what we now call the essentially complete genome. And this is build 35 that many of you use. That was published in 2004, and set some records. Well, first of all, the genome was in good shape. 99% of the U chromatin had only 340 gaps, and the quality was even higher than we thought. But we now had a new record number of 2,700 authors for this paper, showing how many people were involved in the whole program. Now, you can see here who did what. Around 10% of the project was completed in Houston, about the same amount in Department of Energy and in Boston. And much of the work was done at the Sanger Center and also at, in St. Louis by Washington University. And about 16% of the sequence came from all the other labs around the world who had worked through the whole period. Now, at that time, we finished the genome and got it out there and published it. And what did we learn? Well, we certainly learned that the number of genes that were there was less than what we had originally suspected. We learned that the quality was important. That is, the work we put in to make it finished was able to show us many duplications and base errors that were in the draft. Here's an example. I'm sure many of you know what happens when you do an alignment and a base is wrong in the reference. It can lead you to a lot of spurious work. So we avoided much of that, and we learned how to sequence other genomes and manage large data in engaged communities. But we certainly hadn't achieved what we originally set out to, which was how to figure out the genetic basis of disease by comprehensively interpreting this genome. Now, that set us about tackling some new programs. Now, many of those were in the area of comparative genomics, where we sequence other species, but I won't talk about that at all today. But some of those were more poignantly in sequencing other humans to establish rules for studying genetic variation and trying to associate that genetic information with disease. The two premier projects at the time were the haplotype mapping project and the 1,000 Genomes Project, and I'm sure many of you have heard of those. Now, as we were doing this, of course, the technology changed some more, and sequencing by dideoxy terminators that were put through a capillary to give a chromatogram like this suddenly went away. The methods originally conceived way back in 1990 were enabled by a group at Cambridge that did some remarkable work making mutant enzymes and special nucleotides that you use in the Illumina machines of today that have cleavable fluorescent linkers and reversible termination, allowing with cluster amplification to produce spots on a solid surface, allowing you to grow DNA bases on a spot and use them in these recognizable Illumina machines. That happened around the early 2000s and really changed the perspective of things. So those methods were used in this early study of how much variation there was in humans. Now, those projects established a couple of important things that really impacted us in the early 2000s. The first one was we suddenly had a handle on common variation. So I'm depicting here the HapMap project and 1,000 Genome Project that looked at major continental populations around the world, looking for the genetic variation that was there. Here I'm depicting common variation, where early in a population there's a mutation, and of course as the population expands, that's at high frequency because it happened early in the lineage. Here on the right, depicting rare variation, where the mutation doesn't happen till much later, and you see that late in the lineage. So what these projects did is they gave us tools to explore common variation, and they let us know there was actually more rare variation than was originally expected. And this is because the population expanded, a human population expanded quite recently in this, after this long sort of narrow bottleneck. But as far as the common variation goes, this was used in GWAS studies and is still being used in GWAS studies. And this is the 2006 Wellcome Trust Consortium that produced the first really set of Manhattan plots showing genetic association with different diseases in a population that they'd gathered, phenotyped, and subjected to genotyping. Now, unfortunately at that time, although there were many disease associations, there were no causative gene alleles that were yet found. And the short story is, and many of you know, that that's still the case. Now, 2 decades or a decade and a half later, we now have tens of millions of GWAS samples and millions of genome sequences from large programs, including the UK Biobank, Genome England, TopMed. This is the National Institutes of Health, Heart, Lung and Blood program. The Common Centers for Common Disease Genomics, the NHGRI program, and more recently, the All of Us program in the US that combined somewhere between 5 and 10 million genome sequences, plus all those genotypes, which have produced some remarkable science and some really strong disease association, but still a paucity, a shortage of alleles that have real impact on understanding genetics through mechanism. So that's the status of what's happened with common disease studies right now. Now, I depict it here by pointing out that adult genetics is hard, and that this is GWAS, which used the common disease, common variant hypothesis, has been very successful in mapping, but has produced very few actionable alleles to explain all of the genetic burden of disease. Now, the second major technical development that happened in this whole period, a little later on, was the development of DNA capture. The idea that you can use oligonucleotides to extract the 1% of the genome that you might be most interested in because it contains the genes. And these methods, which are used ubiquitously today, began with these trial experiments on solid supports, producing hundreds of genes, not the whole genome yet, then quickly evolved to the whole genome methods and were applied to exome sequencing, just like you see today. Now, that period together launched what we call the era of personal genomes. And we were able to do a lot of this work in Houston. The first of the next generation sequencing projects, actually on 454, not on the Illumina programs, was that of the sequence of James Watson. And here he is getting his sequence given to him on a disk here in Houston. And a little later, we applied the solid WGS methods, these were ligation-based sequencing methods, to sequencing of two key individuals. One is Jim Lubsky, who was Claudia's mentor together with me in Houston during her graduate work. And then a little later, the Beery twins, who were individuals with a genetic disease. Now, the remarkable thing about this is that Jim had been working on his own genetic disease for more than a decade, and had discovered, I should have said, Dr. Lubsky here has a genetic disease. He had become a world's expert on that because he studied his own family and other families. He found the genetic basis of many cases of this disorder, Charcot-Marie-Tooth, but he never found the gene that was missing or deficient in his family. And when we generated a whole genome sequence of Dr. Lubsky, and Claudia here set out to analyze the data, she found the gene that was deficient in him. So this is like a big milestone in personalized genomics. Now, a little later, we did this similar experiment on the Beery twins, and were able to intervene in their medical condition, which was a severe movement disorder, by using the sequence to guide the administration of a common supplement to them that restored their movement completely. So this was our poster child for medical intervention through sequencing. But throughout this period, as we refined the methods and applied them to different genetic disorders, this was one of the first ones, Bainbridge-Roper syndrome, we realized that we could apply this in the clinic. So we moved the methods from the laboratory into the clinical diagnostic and began doing serious clinical sequence. And it's this work that's led us now to where we are today with thousands of genomes produced in Houston. Now, as we did that, we recognized that rather than focusing on the adult disease problem straight away by GWAS and those kind of methods, perhaps if we focus more strongly on Mendelian problems, then we could make more progress in ultimately understanding the molecular basis of human disease. Now, those technologies plus good old family studies really provide us a mechanism to drive that work. Now, if you think about Mendelian disease, you can ask, what is the opportunity and what is the scope of the problem? There are, of course, about 20,000 human genes. Probably a little more than half of them are solvable. That is, if you studied enough people, you would find a way to link mutations in each gene with a human phenotype. Now, there are about 4,700 of those that are solved at this point, but we still have a large fraction, about a third of the genes for which we don't have the linkage between the gene and the phenotype. And our thinking at that time was if we could advance that more quickly, perhaps we could make some real progress in understanding genetic disease through genomics. Now, here's a slide I'd like to show, which is a model for how we were solving Mendelian disease at that time. It's the idea that we would take a family who had what potentially looked like a genetic problem. We would use the new sequencing methods to sequence key individuals and identify different genetic signals that could explain the disorder. The most straightforward of those, of course, is if there is a de novo mutation in the proband. And if you do that in a few families with similar phenotypes and you find the same gene that is mutated, then you indeed have found a linkage between that gene and the phenotypes, and you can declare you have found a new syndrome. In this case, I like showing this slide because this is my grandniece. I'm sorry, this is my grandniece. And my sister had called me way back in 2012 to say that there was some problems with little Molly, her daughter's daughter, and that perhaps there was something we could do in the laboratory to help out. And without really much surety of success, we sequenced the family and it did indeed find a gene, AHTC1, which is mutated and which we now study and have about 400 families for which have this disorder. The problem with these cases is usually that the phenotypes are very general. That is, it's hard to pick based on any clinical features what is the problem, and you have to resort to a molecular test. Now, this general activity has gone on in multiple programs and in multiple countries, up until recently largely driven by the National Institutes of Health, using sequencing methods and family studies to discover new Mendelian diseases. And we have a program here in Houston. I work with Jennifer Posey and with Dr. Lubsky, actually pursuing these genetic diseases. And we find that for about every 30 or so genomes that we sequence with the right families, we make a new gene discovery. And so we're steadily building the Mendelian catalogue. And that got us, that kind of approach has got us from around 3,000 to the number of about 4,700 that we have now. So the question that we have, though, is, will we live long enough to see this catalogue be filled out? Because as you can imagine, that still leaves 4,000. We could probably, what's 4,000? It would take another 20 or 30 years to fill out at the current rate. So we've been looking very hard at other ways to speed up discovery. And because I wanted to spend time having discussion, I took out some of the slides that underscore each of these different methods. But I do want to mention some of the other approaches that we've been using that have been trying to speed up genetic discovery. Of course, we've been trying to do as much sequencing as possible with as many families. But of the data that we have from existing families, which is, we have probably 20,000 unsolved cases, samples from unsolved cases, just here in Houston, and there are many more worldwide. By looking at the computational comparison of the unsolved genomes, it's possible to find a few cases where particular genes are mutated more than expected. And this gives some indication you might have a disease gene and grounds to follow up. Following other omics, RNA studies, RNA expression, tissue-specific expression patterns, probing proteomics or metabolomics have all together given insight into some more of these genetic disorders. They've each been effective in their own way, but none of them have really solved the problem of how do we dramatically accelerate Mendelian discovery. They're best, most of these methods are best if you're looking at sort of known situations. But what we really want to do is speed up the discovery. And it turns out the way to do that is to get more phenotyping data. So that's our big struggle right now. Now, why do we think the phenotyping data is going to be so helpful? Well, Dr. Posey's done some other studies that are relevant here. When we first started doing genetic diagnostics of these Mendelian cases in the clinic, a very surprising discovery is that there were multiple individuals who actually were bearing two genes, two disease traits, two genes that were mutated. So of course, in general Mendelian disease, you have a child, you have one gene mutated, you have one disease. But then there's a fraction of cases, the child might have two different genes mutated. Indeed, that might be why the phenotype is hard to tell, hard to diagnose, and why the child was referred to genetic testing. And the numbers were a bit surprising. Actually, 5%, just about 5% of the initial cohort that came through the diagnostic lab actually had this kind of situation. Many of those, or most of them, had simply two Mendelian diseases, but a few had three, and one individual I mentioned even had 4 Mendelian diseases. Now, as you can imagine, if you have two Mendelian diseases at the same time, they could be in two different categories. You might have two distinct disorders, for example, something that affects the eye, and in that case, you would expect all of the phenotypes to be present without contribution of one gene to the phenotype of the other disorder. But then there'd be overlapping situations where sometimes the disorder would affect the same organ. These are both developmental and epilepsy disorders, and somehow there would be a complex interplay between the phenotypes driven by the two different gene mutations. This would be called overlapping. And what Dr. Posey and your group were able to do was to categorize these two different situations, the idea of the overlapping versus distinct phenotypes, by assigning HPO terms, human phenotype ontology terms, to the different cases that they studied, and then using a computational model to decide whether or not it was possible to digitally distinguish cases of distinct versus overlapping. So the basic idea here was to have a human set of clinical experts categorize the two different situations and score them and compare that to the HPO mapped computational model. And it turns out the computer does pretty well in distinguishing distinct from overlapping phenotypes. And this is very heartening to us because it tells us that at some level we can use a computational model for phenotyping in order to extract phenotype-genotype complexity relationships. It's a big milestone. Now, I just want to give you one quick example of how well this worked. This one individual who had 4 different genes mutated, presented, and Engard and Isabella dissected the phenotypes from this individual and partitioned them out and produced this map. So you see down the bottom here, the different phenotypic spectrum okay, so here's the phenotypes listed across the top. And each of these sets of horizontal marks indicates the phenotypes that would be expected from those individual genes, from the 4 different genes that are mutated. And indeed, when you examine this poor child, then each of these was present, but each was contributed from some of the different genic underlying problem. So again, a powerful indication that you can in fact dissect these phenotypes and then go ahead and use those to build stronger genetic models. Now, where does that leave us all then? Well, it leaves us with this idea that common variation following the Genome Project hasn't quite got us yet. Studies of that haven't quite got us there yet and explain to us what's going on in all of human genetics. But it also says that if we get more clinical integration, we get more phenotyping, then we will be able to make particularly in single gene disorder, important question of how do we get more clinical genomic integration? And we have there what I call the problem, the catch-22 of clinical genomic integration. Basically, genomics is not used much in the clinic yet because it's not useful enough. But the reason it's not useful enough is we don't have all of this clinical information we'd have if we were using genomics in the clinic and integrating that data back into our research. So much of what we're doing now is to try and build models to integrate the clinical work back into genomics. So this doing that. When we decided to try and advance this cause, we thought about all of the different areas that genomics could be most used in the clinic. And that, of course, means cancer, pharmacogenomics, and there are a few others that I'm sure you could list if you were pressed. We decided to study cardiovascular disease risk and use that as a hook to get our clinical colleagues engaged in genomics. We designed a test, it was actually a panel, where we would enroll patients who went into the Baylor clinics. Now, these are not very, very sick people. These are people with mild disease. Basically, get them consented. The physician would see them, would consent the patients, take their blood, sequence them, return a genetic report to them, and then have the physician return to them any information about their genetic status. And then we would use that information in research and discovery. Meanwhile, our big gain would be we've engaged the physician community. Now, we had to do a lot of practical stuff I won't tell you about, but build a whole genetics testing infrastructure. And we enrolled people. We stopped at 600 or so because of COVID. Didn't quite get to our 1,000. And we designed genetics reports that either gave them indication they had a dramatic Mendelian disease-like condition that was carrying through to their adulthood. These are adults, by the way. Or else we gave them other information about polygenic risk, I'm not going to talk about today, unless you ask, and other lipid markers and pharmacogenomics. Now, the really stunning thing about this study, the really exciting discovery of it, was that a large fraction of the individuals who participated actually had Mendelian disease that had not been diagnosed in them until adulthood. And that's this category here. About 65 people had various disorders and pathogenic or likely pathogenic mutations that mark their disease. Overall, nearly 30% of the participants got some genetic information that impacted their clinical care, but 10% of them had really severe conditions. Now, some of them, I'll just a couple of vignettes. Some of them had connective tissue disorders. Here was a case of a person who was diagnosed with Marfan syndrome. After the testing, it was realized they had Loeys-Dietz syndrome, which has quite different clinical follow-up and monitoring, and drug treatments made a big difference to their ongoing therapy. There were several cases of hyperlipidemia, an individual here who from childhood had enormously high lipid levels. We found out that she had an unusual pathogenic variant in the low-density lipid receptor, and her whole family benefited from this hypercholesterolemia diagnosis. But in addition, she was able to be enabled for insurance coverage for a drug that was now believed to be much better and more suitable for her rather than her existing treatment. So there are a lot of these anecdotes that said this stuff can be really useful at the 10% level in these enriched cohorts. That's excited us a lot because it provides us with a gateway into the clinic where we can convince physicians that it's worthwhile for them to do genetic testing, and they can then adapt to the process of genetic testing and allow us to get a research cycle going where we provide testing for clinical patients, but we also are able to access the data for more discovery, which is what we're doing now. Now, this is not all as good as it seems. So just to explain that, if you went out and sequenced people randomly, just on the street, you probably find a little under 1% of them had a disorder or had a Mendelian type signal that said that would affect their health trajectory through their life. It's less than 1%. Now, in this clinical environment we've got here in Houston, we had enriched that to 10%. That was the exciting thing. That made the work worthwhile. But now we're doing a replica study down in the Rio Grande Valley. We find the hit rates are a bit lower. They're probably around 3%. Now, some of the participants in this work are dismayed by that, and they think 3% is very low if you go out and sequence patients, be giving them important genetic information. But the fact is, it's not low. It's it's around the mammogram rate if you take out the false positives. So 3% hit rates is pretty good. It's actually much larger than many other tests. The real problem is it's still a bit too expensive. It's still about $1,000 per shot. And if we could get that down to something much more tractable, I think we would have grounds to push genomics into the clinic and really get a toehold and allow us to gather this information for research and discovery more readily. Now, we need more technical innovations to drive that. And so these technical innovations now are really the key to progress in understanding genetic disease. So those are the totality of my comments. I would say that, what we've shown, we've done the Human Genome Project, we've shown the background rate of nature of human genetic variation and genetic diversity. We've really got a good handle now on Mendelian disease, and we're making big progress there. But if we want to push it further, we need this clinical integration, and we need to drive these assays to bring even more high utility. And I think I retained this one last slide, just to remind you all of what the sort of landscape of the challenges are. All those technical innovations I told you about in this last 50 minutes, really just led us to the current Illumina world. There are at least 10 platforms out there now for DNA sequencing that are under evaluation and have established utility in different areas. And these are the things that we're examining now to reach this idea of the quicker, cheaper, easier, faster test. So lastly, I'll just thank some of the individuals here. This is Donna Musney, who's worked with me for 30 years in the Genome Center, our co-director, other staff and people I mentioned along the way. And I want to thank Claudia very much for inviting me to speak and for the work that she did, or misspelled special, sorry, the work that Claudia, you did while you were here in Houston. So thank you very much for listening. I hope we've got a few minutes for discussion.
Thank you so much, Richard. And if people have any questions, please raise your hand or put them in the chat. I'll stop the recording here.